All right, we're good to go.
Hi, I want to welcome everyone to Darby Creek's Watershed 101. Today we have lunch with Carol talking about microplastics. But first I want to go over a few just topics or housekeeping items. We're going to have the chat room open. If you have any questions during the presentation, you can just type it in, or when we come to the end, we'll have a Q&A. And if it's a long question, I'll ask you to read it. Otherwise, we can just read it for you and Carol can answer it. First, I want to talk about Darby Creek Valley Association. Sorry about that. We are a nonprofit membership-based organization. We started Darby Creek Watershed 101 with Willistown Conservancy Trust and Stroud in this great partnership to help educate people in our area. With the collaboration, we were able to really join our resources and expand our education program. So if you have any questions about Darby Creek at the end, you can visit our website. And for now, I will introduce Carol.
Susan Miller.
Okay, Carol, do you want to share your screen?
Yes, hi everyone. Thanks for coming and thanks for your concern. Oh wait, I have to, let me do this in a different order. Thanks for your concern about microplastics. Can you see it yet? No. Sorry about that. And now it should work. We good?
You're good, Carol.
Okay. Well, thank you again for your interest in this topic. I want to start with a quote from a first grader as taken by the AmeriCorps New Jersey Watershed Ambassador Healy Kardec to show you hopefully where our hopes for the future are going. So this first grader was asked what pollution was, and he or she said, pollution is stuff like trash that someone throws out their car on the ground, and then when it rains, that trash moves and goes into the drains and rivers and then travels to the ocean, where animals like turtles and fish and whales think the trash is food and will eat it and get sick and die. So they've made all of those connections, which is really a great testament to our environmental educators. So here is an outline. Okay, so I now have run into the issue of which, let's see, which key will advance my slides for me. Are you hearing my computer complaining? Oh, I just have to click it. All right, here's the outline of what we're doing today. We're going to talk about the sources of microplastics, the methods of their release into aquatic environments, an assessment that was done by a review of methods and research methods and outcomes, as well as the recommendations as a result of that review. We'll look at the effects of wastewater treatment plants, biofilms on microplastics, also called plastospheres, microplastics in sediments at the bottom of streams, effects in aquatic organisms and the food web, and then we'll end with studies in Pennsylvania streams that are still emerging and talk a little bit about social consequences. Perhaps we should be talking about that. So 60 years after the entrance of plastics, let's see, actually it's 70 years after the entrance of plastics into our environment, this is the reality that no one anticipated. At the bottom two photos, they are from the tidal island at the John Hines National Wildlife Refuge, where Darby Creek and floods into Cobbs Creek, floods into Darby Creek, which floods into Tinicum Marsh, and then down into the Delaware estuary, and then the ocean. And what you see at low tide is on the left, it's a Phragmites field with all of the planty debris on the ground. And on the right, with red dots, are all of the plastics that are embedded in that field. They're very difficult to see, and you wouldn't even know they were there if I hadn't been there and saw them. And you see that they have all taken on the color of the debris around them, which is an obvious an obvious example of how plastics will, how other material in the area around the plastics will absorb and attach themselves to the plastics. We have a high dependency on fresh waters for drinking water and food sources, and we need to understand our distribution of both macro and microplastics in our streams and rivers if we're going to develop appropriate policy and management tools like TMDLs. The environment is already committed to hundreds of millions of tons of plastics in the ocean, and we are cumulatively increasing the tons of plastics increased into the environment every year. What I mean is we are increasing arithmetically the tons of plastics released into the environment every year. Every year, a larger amount goes in. More than 50% of plastics are meant for single use, and since 1950, about 50% of all plastics was landfilled or dumped into the wild. Jan Beck, who made an attempt to estimate the actual amounts, estimated, Jan Beck et al., estimated that in 2010, the world produced, in that year alone, the world produced 303 million tons of plastic waste. This is only increasing over time, and it's increased further in the time of COVID. They estimated, Jan Beck et al. Estimated that 5.3 to 14 million US tons, and today when I talk about tons, I've converted everything into US tons. They estimated that 5.3 to 14 million tons made its way into the oceans just in 2010. Plastic recycling has plateaued at 8% for a number of years, and it even decreased in recent years according to the Pennsylvania Department of Environmental Protection municipal waste reports. Jan Beck et al. Estimated that the cumulative quantity of plastic waste that can enter the ocean from land will increase by an order of magnitude by 2025. So just briefly, what are microplastics? They're generally defined as less than 5 millimeters in diameter. They are made up of microbeads, that is things produced as microplastics, as well as fragments of larger items broken down in the environment. Usually these categories are used: microplastic films, microbeads, microplastic microfibers, microplastic pellets and nurdles, lines, and foams. We have a high dependency on fresh waters for drinking water and food sources, and we need to understand our distribution of these microplastics. Okay, I'm sorry about that. As recently as 2012, awareness was still relatively low. So although microplastics have been around for a long time, it's only recently that scientists and the public have become aware of just how serious the problem is. The background of this slide is from the United States Geological Survey, the USGS. They were measuring wet deposition in the Rocky Mountains in remote areas higher than as high as more than 10,000 feet. And they found they were measuring just wetness. That was the purpose of the study. They were measuring the water that came from the sky in any form into their collection buckets. And they incidentally found plastic particles in more than 90% of their samples. And here is a variety of what they found. Their project in 2019, or their paper, was titled, "It is Raining Plastic." So we're going to talk about the sources of microplastics that get into our waters. Here is an example of what was picked up in a wetland and shore area at Barnegat Bay Partnership. First, there are the primary sources. So these are things that are made for use. So microbeads have been in personal care and cosmetic products since 1950. Pennsylvania banned them in 2015 from being produced. And in 2017, they can no longer be sold in stores. However, China is still producing them, and they have finally banned their production beginning in December of this year, but they can continue to sell existing products until December 31st of 2022. But please note that, so depending on where you get your products from, you need to look and see there could be microplastics in them. Also, they are still being used in industrial and home laundry detergent. So again, you have to look at ingredients and look for items that could be plastics. They are still allowed to be produced in laundry detergent. Also, there are production pellets. These are polymer resins, so this is kind of like virgin plastic that is shipped around the country and around the world to manufacture plastic products. Let's see. So then there are air blasting technologies that use these microbeads as an abrasive method. Then the secondary sources are fragmentations of larger plastics by biodegradation, which is breakdown by microorganisms like bacteria and fungi. There is photolysis, which is plastics broken down by light and ultraviolet energy. There's thermooxidation, which is plastics broken down by chemical reactions. They reduce the molecular weight of the plastics and they lose their intended functions. And then thermodegradation is plastics broken down by overheating. And again, the long chains of plastics, the longer the chain, by the way, the stronger the plastic. These long polymer chains, monomer chains to break down, and it also changes the functions of the plastic and the properties of the plastic. Then washing synthetic textiles has become a huge issue. It's thought to be, we're not sure yet, but right now we think it's the largest source of microplastics in waters. We're going to see today that microfibers are the largest type of microplastic in our fresh waters. There is a product called Lint LUV-R filter. A study compared two commercial lint remover products and found that this one, LUV-R, removed 87% of the microfibers by count in washing effluent where fleece blankets were washed. Just by comparison, the other one, Cora Ball, in case anyone has been looking for these things, that only decreased by 26% the amount. Then there's waste and drinking water treatment plants and the combined sewer overflows that will put plastics in the water. So waste and drinking water treatment plants to some extent filter out microplastics, but not perfectly and not all parts of their production. But the combined sewer overflows, that's water from land that never gets to the treatment plant and flows directly into the streams. So rivers are have particularly important roles in global microplastics. First, rivers provide, in terms of just our water use, part of what rivers serve many important ecological services, but just in terms of water, they provide our drinking water, our irrigation, our wildlife habitat and biodiversity, and our recreation. These are all ways in which plastics might change the value of those things. There are greater concentrations in streams than in bays and marine environments. So this is just preliminary information on this comparison, but this is an important study that McCormick, Hoellen, and Mason and others in 2014 did. And they were interested in the bacterial assemblages on plastics, and I'll show that later. But right now, what they did is they looked at studies of other marine environments and water environments that use the same research methods that they used, so that they felt they could make an approximate comparison. And they found, looking at the graph, they found that upstream of the water in their study around Chicago, there was a relatively low level of microplastics. Downstream of a wastewater treatment plant, there was a high level. And all these other environments, the Great Lakes, two California coastal areas, the North Pacific Gyre, which is where the Great Pacific garbage patch is, and these two other North Pacific ocean settings all seem to have lower levels than they found in the river that they were studying. This is supported, by the way, by a more recent study by Cohen et al. In 2019 at University of Delaware. They found higher concentrations in the upper Delaware Bay than in the lower Delaware Bay. The smaller spatial space available was one of the reasons why there might be more, and that's what McCormick et al. Postulated that that rivers will have the same sources perhaps of microplastic pollution as marine areas, but they will have less water volume for dilution and may have higher microplastic concentrations. So the University of Delaware kind of supports that, although there are other variables going on in the bay, buoyancy and wind and tides, other factors that will affect how much the concentration of microplastics in different parts of an environment. The POPs are persistent organic pollutants. These are pollutants that are not biodegraded, and they're not broken down by organisms. Their concentration in living organisms in the river is much more than the concentration in the water around the plastics. So the pollutants will attach themselves to plastics in a much higher concentration than what is in the water around them. And because of this, there is a bioaccumulation of these pollutants because we know that organic organisms ingest microplastics and that microplastics impair their reproduction, change their development, cause malnutrition, cause internal abrasions, blockages, and possibly death. Furthermore, we also have direct evidence now that toxic chemicals, such as gases and also gases that are in the plastics due to their production methods, can leach or exude into water. So this is how they are being exposed. And I'll show you a study that suggests it's even more than it's more from the plastics themselves than from what's in the water. So the bioaccumulation is an issue of gradual and increasing accumulation of these pollutants and contaminants in organisms, because the organism absorbs the contaminant faster than it can break it down. It can catabolize it or metabolize it. So that's thought to be why they are bioaccumulating, or that's a principle in biology. So what are the modes of microplastic release in aquatic environments? So I'm going to give you some, I'm beginning to tell you about a very helpful review, El Khatib and Ovana Del Craver from the University of Rhode Island. Some of this information comes from them and from the studies that they reviewed. So first of all, wastewater treatment plant effluents are known to increase microplastics in streams. They are one of the main sources of microplastics. They can remove, at least in the United States, 50 to 98% of microplastics by skimming and sedimentation processes, as well as by some other more involved secondary and tertiary treatments. The problem is that there are such high volumes of effluent from the treatment plant that are discharged daily. So even with relatively low amounts in the water, if it's a very good treatment plant, if it's an updated one, and they're very expensive to update, if there's still such large volumes of water coming out. Also, there is the overflows, the combined sewer overflows that discharge without treatment and carry a large amount of plastics. The wastewater treatment plants, also the microplastics that they remove from the water, they go into biosolid sludge, which has long been used as a fertilizer on farmland. About 50% of the biosolid sludge that comes from a treatment plant is reused on farmland. And a study by Nozzetto et al. Found that it remains in that soil for decades. These authors estimated that 44,000 to 300,000 microplastic tons per year are put on the soil in North America and even more in Europe. Although wastewater treatment plant biosludge is treated before it's reused, microplastics are not targeted for removal. Then 5 to 10% of microplastics in our water come from tire wear and road markings that are quite small, 5 to 25 microns. Also, industrial fluids are often used in open space. These are, and then building coatings are also in open space, and they have the ability then to escape by air and water into the stream. These industrial fluids I'm talking about are things like drilling fluids, rust and paint removers, cementing paste, and things like that. Then there's the unregulated disposal of plastic products in our streams. Many people have the experience of seeing illegal dumping. And then landfills are areas that have many conditions that will degrade plastics, including ultraviolet light, high temperatures, acidity or low pH, salinity, microbes, and mechanical stress. All of these conditions will break plastics down into microplastics. And then there's atmospheric deposition, including rain. So there are now many studies showing that the microplastics are coming down every day from the atmosphere.
More plastic container. We shouldn't buy any more plastic containers.
Then there's another issue about how they're released is the spatial issue from where. So microplastic amounts, concentrations in water per volume do correlate with urban centers. So downstream from an urban center, there is more, but there's a question of land use. Is it the land use itself? What about that? So another study is going to get to that down the road a little bit. So we need more understanding of the sources, the accumulation sites, and the movement of microplastics in rivers in order to quantify our global microplastic distribution as well as for our local purposes. So this is one study of the Great Lakes back in 2013, which they looked at microplastics in the Great Lakes. They looked at surface water samples that they defined as 1 meter down. They actually only measured them in Lake Superior. Well, this is information that's combined from that study from both Erickson's paper and Sherry Mason's talk at the Stroud Water Research Center in 2019. And so from Lake Superior in Huron at that time, and this is going on 10 years now because it wasn't published in until 2013. There were 7,000 microplastic particles per square kilometer in both Lake Superior and Lake Huron. In Lake Erie, there were 47,000 per square kilometer. In Lake Ontario, there were 230,000 per square kilometer. They said there are similar garbage patches in the Great Lakes. These lakes have a coast that's longer than the east coast of the United States, and so that puts them in great contact with human activity. The types of plastics that were most common in that study were fragments and pellets. They did not track microfibers in that study. They did not report it. Then a second study, also out of Mason's lab, published in 2016, looked at 29 tributaries into the Great Lakes. And those 29 tributaries accounted for about 22% of the total tributary contribution to the Great Lakes. And in this case, where they did track microfibers, fibers were the most common. And they found that for all of the types of forms of plastic, the fragments, films, foams, lines, pellets, and fibers, of all of those, everything but the fibers had greater concentrations during a storm runoff event. So once the water flushed off the land into the stream, except microfibers. So microfibers are high in the stream all the time. Let's see. So there are some efforts. So this problem of the Great Lakes has been leading to efforts to try to use satellite technology to try to track where plastics are. More than 10 years ago, there was an effort, COMDEV, didn't work. Now there's another one that's being tried involving NOAA. And I just want to say that we can read about these things, but these things are a long, long way off from finding out if they might be feasible. And so we can't put our hopes in that. So I want to talk a little bit more about research methods. I hope this will be useful. So I want to talk about the review studies findings of the problems of variability in sampling methods and identification techniques. This is from a critical review of extraction and identification methods of microplastics in wastewater and drinking water. This is from the, well, I'll show you in a second. So fresh waters differ from marine water in its chemical components, its organic contents, its surface area, the sources of microplastics, and the sizes of the microplastics. So there are enough differences here that we might need techniques specifically for freshwater aquatic environments. So El Khatib and Oyana Del Craver of University of Rhode Island did this review study. They used 8 databases to find 130 studies, and they used the studies to look at the comparative methodologies in fresh and wastewater, at current gaps and limitations in methods, and they made a ranking system of how well everything was reported, the research was reported. They looked specifically, they compared specifically reported sample collection, sample processing, identification and characterization of the microplastics, how well they reported their quality assurance and quality control methods, and then how they reported out results. So some of the highlights of the comparisons of methods that they found were that here on the left is a sieve method. These are multiple sieves through which the water flows. And then this is a Newston net, which was in my first photo that students might be trained in. They found that in their 130 studies, grab samples were used in 40% of the studies. An extraction pump was used in 20%. Composite sample was used in 20%. Newston net was only used in 12%. And there was a custom method used in 8%. Mesh size and method of identification can lead to underestimating smaller microplastic particles and their actual concentrations. And the thoroughness of wastewater treatment plants in removing these microplastic also varies. And that alone can lead to differences in the concentrations. But this is a major issue that we need to have uniformity on, which is how small of microplastics are they measuring. Processing, people use NOAA's catalytic digestion and separation, and then filtering and centrifugation and staining are all used as part of processing of the samples. And one problem is they might not differentiate microplastic particles from organic particles. And to this end, there is a study, major study being done by the Southern California Coastal Water Research Project Authority involving 36 laboratories in 7 countries. And there are the Pennsylvania State University is one of the participants in this. And the purpose of this study is to develop detailed standard operating procedures for the analysis aspect. I'm sorry, I'm going to go back here. For the analysis aspect. They are looking, their concern is this, in fact, this last point, which is the difficulty of differentiating microplastics from organic particles. And so they have made huge investments to make sure that everything that's involved in the laboratory process is plastic free, including how they clean up the materials, the clothes they wear, all of the tools and instruments all have to be plastic free, a very significant investment. They want to come up with the most precise measurement of microplastics as possible. They are open, so Odette Mina, who is the managing director of Energy and Environmental Sustainability Laboratories at Penn State, in a webinar yesterday, welcomed people to contact her if they are interested in discussing research methods for microplastic studies. If you are interested, we can put her contact information in the chat box. She did urge people to contact her before starting a study because I guess it makes quite a big difference in how you prepare yourself and whether you're going to find the information that you're hoping to find. So if even one person puts in the chat box an interest in it, her contact information will be put in there. So another highlight was that just what this I just referred to, that plastic filters, plastic sieves, fabrics that you're wearing or using, plastic containers, plastic tools, and airborne microplastic contamination are all potential sources confounding your study results. They looked at, this review study looked at differences in how microplastics are identified and quantified. They noted that people were using either Fourier transform infrared spectroscopy, Raman spectroscopy, scanning electron microscopy, or visual inspection of samples. And they also noted that reporting out particles were reported per volume or per mass or per area or per concentration. And there, have some recommendations on that we need to have consistency and uniformity in how things are reported out so that we can make these comparisons. And another thing that differs among studies is some will analyze the whole sample and others will analyze a subset of the sample and extrapolate to the total. Only the spectroscopy methods can identify the actual composition of polymers, but when people use that, well, they tended to use only a small part of the samples and then extrapolated. They noted that quality control and identification methods were the categories that were most underreported. So now some of the recommendations from that study. One, that grab samples seem to be most convenient and appropriate, but you have to use glass containers and they have to be sealed on site. They also recommended stack of sieves with graduated mesh sizes between 20 and 1,000 microns in a laminar flow chamber in the lab. They suggested using the NOAA technique for digesting the water and then filtrating it through 10 and 1 micron membrane fibers for smaller microplastics. And the filters are going to be chosen, though, depending on the technique that you're using for identifying the microplastics. They've recommended Raman spectroscopy with automated software. Now, the automated software is important because it allows, this is very intensive quantitative analysis of the samples, and so the automated software helps it to go a little bit faster, which would allow you to use perhaps more of the water. Or something more recent, the atomic force microscopy, AFMIR. Also, they recommend using the uniformity in units and terminologies, such as microplastics per volume, total microplastics per day in both influent and effluent from a wastewater treatment plant. Not all, those comparisons weren't always made. They recommend identifying the types of polymers and classifying the shapes to identify the sources. So the more we know about the shapes and of course the types of polymers, it may help us with sourcing the material. Also, I want to mention the US Environmental Protection Agency, EPA report. They have a report that identified and prioritized the scientific information needed to understand the risks associated with microplastics in the United States, including methods for assessment, identification of sources and fate of microplastics, risks to human health, risks to ecological health for microplastics. And they also established work priorities and they recognized deficits and needs. So here from the review study, this is 130 studies globally, though it's from around the world. So this is just a looking at the mean comparisons of the types of plastics that were found around the world. They converted everything into the same quantity so that they could look at this. However, what I've learned from doing this is, looking into this question, is that the types of plastics and the amounts are going to vary greatly depending on the human use around the stream. But in any case, this is what they found globally. First is polyester with an average of about 41.5% of the microplastics that were found. And I just want to point out that fourth and fifth, also in pink, nylon and polyamide and acrylic, all of these pink items are plastics that are used to make fibers. Polypropylene, oh yeah, that's in there. Well, polypropylene also is used in textiles, including carpets. And the transportation manufacturing industry is the major consumer of these synthetic fibers, accounting for 35% of polyamide consumption. So we, I think, still have to look beyond our washing machines for where all these fibers are coming from. And then second and third were polyethylene terephthalate and polyethylene for a total of more than 50%, well, for a total of 27% and 26%. Polyethylene is the largest produced chemical used in making plastics. It's more than 60% of the raw material. It's used for many, many applications: bottles, wraps, films, bag, microbeads, etc. Then there's also alkyds. Alkyds are used at 8%. Alkyds are in highway markings, concrete floors, swimming pool paints, marine coatings, insulation varnishes, and so on. And then polystyrene is used to protect things like CD cases, packaging, many types of packaging, trays, egg cartons, buoys, floats, and so on. Here from the review study are the morphologies, the forms of the microplastics from wastewater treatment plants. So again, you see that 51% were microfibers, and they ranged in the studies from 18 to 80%. This is all per meter squared per volume of water. Fragments and flakes were about 30%, also with a wide range of prevalence in streams. Films were about 5%, pellets about 2%, beads 1.5%, and foams about 1%. Now, this presentation today does not address drinking water or drinking water treatment plants. There were 4 studies that this University of Rhode Island group reviewed that assessed microplastics in bottled water, and 3 studies that assessed them in tap water and drinking water treatment plants. In these 7, there were large variations in concentrations of microplastics, and single-use bottled water had higher concentrations than tap water. Other studies have found this too. And there were variations, though, sometimes in orders of magnitude, and also variations in concentrations even within a study. So they didn't feel they could make any outcomes or they couldn't make any comparisons to tell us more about drinking water and drinking water plants. However, only the drinking water treatment plant studies reported polyvinyl chloride, PVC, in the studies in both tap water and in the drinking water treatment plants. Also in this review, the volumes of drinking water were generally very low, yielding the very wide range of concentrations due to various other methodology issues. Concentrations of microplastics in drinking water were higher than in wastewater, mainly due to the size of the microplastics that were analyzed. So that's going to be, again, an ongoing issue. So here is now a comparison of concentration of microplastics in the influence, the water coming into wastewater treatment plants, and the effluents that coming out. Here's an example from a wastewater treatment plant study that I reported from a different study by Free, Jensen, Mason et al. In 2014. And again, looking at the review study, the influence are definitely greater than the effluents. So they range from 1.5 times 10 to the 4th to 9.1 times 10 to the 5th microplastics per meter squared. Whereas then the treated effluents, it was reduced to 8 to 10 to the 1 times 10 to the 1 to 5 times 10 to the 2 microplastics in drinking water. So you can just see that it's much reduced amount of it. However, the influence also had larger microplastics, and this is from a student paper, Sanchez and Ellick, who is from a municipal treatment system. Together they did this study at Susquehanna University. And she found that, yes, larger microplastics were in the influence, and the effluents had smaller and more and only microfibers in the effluents. So again, microfibers are very difficult for water treatment plants to remove, and they are coming into the system in great numbers. And because some of their sizes are so small, they're being missed in some cases. Let me go back for a second. Well, there is another question of, this is just, I've taken this in and out of this presentation. I'm just going to leave it in. So at Rutgers University, 2-day conference in 2019 called Microplastics in Urban Environments, the Camden City Municipal Utilities Director reported that there was more microplastics in the effluent than in the influent. I'm not sure, unfortunately, he didn't give a lot of detail, and I haven't contacted him for more information, but when asked why more was coming out of the plant, he said, perhaps he speculated that it was because the equipment that processes the water is made up of many plastic parts. And there's another study in another part of the world where they also found that the smallest microplastics were the least removed from the water treatment plants. And I'm sorry, this is a 2019 study that found that perhaps the microplastics were getting into drinking water supply chain because of abrasion of plastic equipment used during water purification or during transport. So this is another aspect to it that we know least about. So now talking about plastospheres, so that's just another word for the diverse community of materials that adsorb onto microplastics for some reason. Well, because these contaminants don't mix with water, because plastics don't mix with water, the contaminants adhere themselves, even indent themselves into the microplastics. It's called adsorption. And these contaminants include bacteria, including bacteria that produce enzymes that degrade plastic particles and bonds, cyanobacteria, contaminants from the ambient water that adsorb and concentrate onto microplastics more than what's in the water. Many studies have shown this. These include things like PCBs, polychlorinated biphenyls. They cause developmental and neurological problems. PAHs, which occur naturally in coal, crude oil, and gasoline when they're produced and burned. They are thought to have low toxicity, but they are associated with cancer. Organochlorine pesticides that were causing population declines in fish-eating raptors that have been banned now, as well as some metals, metals dangerous to health like cadmium, chromium, and lead. There are also organisms like hydroids, diatoms, and fungi that will stick onto the plastospheres, onto the microplastics. Even invertebrates, large and small, will attach themselves to the plastics and nutrients. And this is an interesting photo from a study by Schlunt et al., published 2020, looking at the spatial structure in the plastosphere. And they use some innovative imaging techniques. And what they did is they took polyethylene, plastic microparticles, which are the black background that you're looking at. And they put them into the North Atlantic Ocean near Woods Hole. And after 1 week over here, they found that there was various bacteria that had begun to adsorb onto the plastic. By week 2, they found these oval shapes are diatoms attaching themselves. By week 3, they began to see unidentified organic material as well as more bacteria. And by week 5, there was an even more complex array of materials that were adsorbing onto the plastics. I thought it was a wonderful visualization of the problem. This is the 2014 study around Chicago area, McCormick, Coellen, and Mason et al. And they were interested in what kinds of bacteria were adsorbing onto the microplastics. Excuse me, sorry about that. Let's go back here, okay. So these 3 graphs are looking at 4 sources of water down at the bottom here. So the blue column is water that they sampled upstream from a wastewater treatment plant. Then the green is water that they examined downstream from the wastewater treatment plant. They also looked at organic material downstream of the treatment plant and microplastics downstream of the water treatment plant. And they looked at the bacteria in all of these forms of samples, and they used various techniques to analyze what the bacteria were. And I can't tell you in specific about these, but they are methods to look at the richness of the bacteria, the biodiversity of the bacteria, how many different families of bacteria, assemblages of bacteria. So if we look at just this middle one here, they find that, first of all, there is extensive microbial colonization on plastics as well as organic material. Just my computer is slow in thinking. Sorry, it's gonna have, there it goes.
Hey, Carol, this is
Yeah.
Dave. Just wanted to chime in here. We are at about 10 till 1, just to update you on timing here.
Well, can I keep going and people can drop off as they want, or what?
That's a question for Sue, I guess.
I have no problems with that, but I don't know if you want to see, I mean, I asked a couple questions, but you can ignore mine. But if somebody
Oh,
else,
I see. I'll try to go through this faster. So there were significant differences in families of bacteria. There were more families downstream, some more types of bacteria downstream from the treatment plants. There was a greater diversity of bacteria downstream, but also on microplastic films in particular, there was a less diverse form of bacteria on them. This was also found, supported in another study. They basically concluded that the microplastic surfaces that select for a unique suite of bacteria, and that there are bacteria that have an affinity for microplastic, and some of them were pathogenic. So this is now a study done on the St. Lawrence River. This is the Great Lakes down at this end. This is the St. Lawrence Bay up here. Here we have Quebec City and Montreal. These are polyethylene fibers that were, microplastic beads that were found in the water. At all the red places is where they found them, at the blue place, in the sediment. I'm sorry, I'm rushing and now I'm getting ahead of myself. We're going to talk about the microplastics and how they get into the sediment of streams. That's of concern. So the rapid collection of microplastics on biofilms can cause the microplastics in the water to be less buoyant and therefore to sink into the sediments where they may accumulate. The microplastic accumulation, they accumulate concentrations in sediments that are 100 times greater than in the water, and even more, 250 times greater in the soil downwind from plants and in habitats at water treatment plant sites in the sediment. Now, another issue is fecal matter coming from the smallest to the largest aquatic life forms in the stream. They are eating these microplastics, and it causes some of the plastics to go into their fecal matter, and their fecal matter drop into the sediment. So that's another way it gets in there. And then some plastics are more dense, like PVC, and because they are more dense, they are found more commonly in sediments. So effects in aquatic species. There are some different studies. First is just looking at what's in the fish that you take out, or the other life forms that you take out of the stream. We know that they're in their gills and digestive tracts, but a study done more recently by Karami, published 2017, they found it in the edible flesh. They looked at 30 samples of 4 commonly consumed fish, dried fish. They looked at not just the organs that were removed, but they looked at the fish flesh itself. In 2 of the species, they found microplastics were 4 times more in the flesh than in the organs. I'll skip what the common ones were. There are certain common plastics, polypropylene, polyethylene, that are less dense. They float more, but they also are types of plastic that get more biofilms on them, and so they distribute more through the water column. So let me go on from there. We seem to have gone backwards. This is the percentage they found of fragments versus film versus filaments. Then there are these in vitro controlled studies. I do want to go through them. So they found that microplastics will pass through the gut and accumulate in the lipid, the fats storage in water fleas. They found that the chemicals from plastics accumulated lugworms. So this is a study done in the lab where they put the lugworms food prey as well as plastics with biofilms and plastics without biofilms into the water, and they found that the lugworms accumulated more of the chemicals that come from those plastics at higher rates in their tissues because they were eating them in the sediments than in their body walls. So they were in their stomachs more than in their body walls. Lugworms were exposed to PVC without biofilms on them and PVC with biofilms on them, and they found that they burrowed less, fed less, became sicker, and had higher death rates. And the authors were concerned that sediment with more than 5% microplastics by mass could disrupt health and biodiversity in streams. This is now going on to the effects on aquatic organisms. I guess I have to really shorten this greatly, so I'll just put all this out there. Basically, there is a scientist, Brian Pachkowski, from the New Jersey Department of Environmental Protection, who notes that organisms can absorb, distribute, metabolize, and excrete plastic. So that while we don't have that many studies giving direct evidence yet, we know that there's the potential for this. And this is his cartoon showing how it can, even though microplastics cannot pass through cell walls, they're too large, they can pass through the wall of the intestines and go into an immune area where they can affect these immune cells, dendritic cells. They alarm our immune system, and they can go into our lymph nodes, and there is a study showing that that has occurred. In the laboratory, the copepods who were presented, these small crustaceans, who were presented with their food prey as well as varied plastics, they readily ingested the plastics, but then they learned they didn't like some of the plastics. I guess they could tell that they weren't being, getting what they needed from it, and they learned to avoid the natural prey of the same size and shape. And so they started rejecting some of the algae and prey that they would eat, and thus restricted the number of species that they would eat. So anyway, there are mechanisms for both micro and nanoplastics to enter lungs, gut, lymph, liver, brain, and muscle. In food webs, we know that when they're eaten, contaminants from the microplastics leach. They translocate from the digestive to the circulatory system and remain for 48 days in muscles. These are images of microplastics found in mollusks in a study in China in the tissue of the mollusks. It decreases the amount of algae that can be consumed, and they transfer from prey to predator. So they looked at seal scats. They don't eat these microplastics in the water. They eat prey that would consume them. And so I'm just showing that the seals not only have to worry about strangulation by plastics, but from the food that they're eating. So it's in the food web. Now we're going on to rivers in Pennsylvania. And this first study has reported out, this is a study by the Pennsylvania Department of Environmental Protection, Pennsylvania Fish and Boat Commission, and Susquehanna University, because there's a concern about the decreasing population of smallmouth bass in Pennsylvania rivers. They looked at 2 sites in Susquehanna River and in Pine Creek, and they found over 3 years that the number of microplastics in the fish increased dramatically in the 3 years. In 2017, 87% of the fish of the smallmouth bass had 2.3 microplastics per fish. The next year, 95% of the fish had them at 6.1 per fish. And by 2019, all of them had it at 28.9 per fish. And some of the possible explanations are that there's more water going by the fish every year. There's increasing flow rate, as those of us who live in the area know, each year. There's probably an increasing accumulation of plastics from human cause, including increasing amounts being put in the environment, as well as the increasing accumulation from legacy plastics. And then the smallmouth bass also eat large variety of prey that lead to many pathways for accumulation. So I'm at the end here. Just quickly, some of the social consequences. This is a photo of Darby Creek with large plastics floating in it. At sites where commercial debris is apparent, it reduces the intrinsic value of the stream to visitors. And people who live are more likely to litter if the habitat is deemed less important. So this is also an environmental justice issue. Poorer neighborhoods that are deemed less important might accumulate more litter based on the psychology of how people think. Waste attracts more waste. Stream debris is not correlated with land use, except that in the most urbanized areas that do have higher densities, but the intensity of human activity in a riparian zone does affect the amount. And this equates to increasing human population around our streams is going to lead to more waste. And also the point that because streams are smaller, there's a more connection of the riparian or the river edge area to land, making them more in contact with that human activity. So thank you to the Darby Creek Valley Association. Thank you to the Hines National Wildlife Refuge, rangers and volunteers who pick up plastics in Darby Creek. Thank you to the Stroud Water Research Center. I will unshare so I
Okay.
can look.
Thank you, Carol. That was very informative. Right now, I want to open it up to any questions. Does anyone have any questions? You can either type them in or you can unmute yourself. Does anyone have any questions? Well, I'm going to ask my question. So with the microplastics in farm soils, is there any chemicals that leach into the soil and then back into the food as it grows?
Right, so that's a really important question. Also, how much of the plastic is in plants? When I first looked at this, there was no firm evidence. There was one student that found that the roots were not taking it up, but the webinar I heard yesterday, the field scientist who also presented from Penn State, Lisa Emily, who's doing microplastic studies in the western part of the state, said that the plants might be taking it up. So we're at such an early stage with this. The amount of literature has grown, explosively over a very short period of time. So that's a very critical question, but we're just trying to find out if the plastics are in the soil and in the plants themselves.
Thank you. And now with the microplastics in soils, and I don't mean like with the farm soils, but in other soils, how does the microplastic particle size change the soil type to behave? Is it more like a sand if it's mixed in? Like on a stream bank, would the stream bank act more like if it was originally a clay with the microplastics going into it, change its characteristics to be more like a sandy type of soil?
Yes, I don't know. I don't know enough at all to begin to speculate about that, but I will say that Lisa Emily speculated on that yesterday, saying, well, it could make it more slippery because remember, plastics are made out of oil and gas. I don't know long-term how it's going to change our banks. We don't know whether it's going to make them degrade more or not. We know that they're stuck in the banks. I see them all the time, but we're not able to know how they're going to change the soil visibly to our perceptual level.
Okay. Has there been any engineering studies on, like from the Army Corps or anything like that, on compaction with soils when building?
Oh, what an interesting question. Yeah, I don't know. And I would just ask Dr. McGillivray, if you want to, say something that would be really helpful about any of these questions. Just please speak up if you like.
Yeah, I don't think there's information to inform those questions really at this point. I will, if I knew anything, I would add information, but
yeah,
I don't think so.
I mean, this is pretty, these studies are very impressive, but these are just the beginnings. Trying to get a handle, and one of the things I take away is that each stream is going to give you different results. It's going to give you different results on what the nature of the land activity was around it and the nature of the methods that were used. So there is not a one-size-fit-all outcomes when it comes to our streams.
Okay. We have another question. A few words more about biodegradation in soils and aquatic environments. What could the MPs be degraded to?
I know Dr. McGillivray would be more qualified than I to say something about that.
Well, I would just say that plastics cannot be biodegraded to truly mineralized to like water and CO2. Plastics can degrade to smaller, they're polymers, so they can degrade to smaller polymers, but they cannot be completely biodegraded.
Right, and so the size of the particles that your study is able to identify becomes very important, is very important.
Okay. Does anyone else have a question? Okay, if nobody else has any questions, I don't see anyone if they raised a hand or not, but I want to thank Carol. It was very informative. You did a great job. Oh, I have one more question. Do rocks, pebbles filter any of the MPs? I would say no, but
Rocks,
the expert.
rocks, what?
Do rocks, pebbles filter
Filter.
any of the microplastics?
No, unfortunately, what they're going to do is to break them down into smaller microplastics. They're just going to make it more available to smaller and smaller organisms and more difficult to identify. With the idea, those couple of factors that I reviewed of how those very tiny microplastics might end up in the soil.
Okay. All right. Well, Carol, once again, I want to thank you. It was a great presentation. For anyone that's interested, we did have this recorded, and I will send out a link for you to watch it if you want to rewatch it. And,
Yes, I'm sorry. I do need to mention a couple of other things. Sorry about this. I learned this from Dr. McGillivray. One, you all, I think, would be interested in this. The USGS and the National Park Service are doing a comparison study on Lake Mead out west, sorry about that, and the Delaware River in Pennsylvania. And they reported out their result on the microplastics. They reported out their results from Lake Mead. They have not reported out the results from Delaware, to the Delaware River. But also, Dr. McGillivray is heading a study looking, oh, I guess I may have mentioned it, but anyway, looking at the microplastics, yes, he mentioned it, in a very heavily used part of the Delaware River. I know the challenges are going to be huge for that, and we're going to be very grateful for the results of that study when it's done. So those are things that are up and coming.
all right. Thank you very much.
Thank you.
So if nobody else has any questions, Carol, I want to thank you again. It was a wonderful presentation. Hoping that maybe in the future you might be able to do another one for us. It was a great job. And thank you very much.
All right, thank you all.
All right, and
Have a
just to
great
let you know, before we sign off, we do have more webinars coming up, so you can check DCBA's website and sign up. You might, because you already signed up for this one, get emails in regards to more presentations. So thank you very much.